Okay, seems like we have almost everybody on. So, hello everybody. Thank you so much today for coming and attending the second part of our Benchling Bootcamp series. Today, we are going to be covering the basics of our molecular biology suite. So, quick agenda for today. We're going to spend the first few minutes going over some welcome and introductions, then a brief overview of the Benchling infrastructure and our molecular biology suite. Then we'll spend about 40 minutes within the platform itself, going through a sample use case and demo. And then we'll spend the last few minutes going over recent product updates, as well as resources and next steps for your team to access. Okay, so some quick introductions. My name is Priyanka. I am a training coordinator here at Benchling. On the call, we also have Diana, who is our support specialist, Chin, who is our training manager, Stefan, who is our implementation manager, and Meredith, who is our customer success manager. And they're going to be here on the call to provide additional support as we need. Okay, so just as a reminder to everybody, this is supposed to be an interactive session. So at the bottom right-hand corner of your screen, you should see this Q&A button. And this is where you can type questions directly into the chat if you'd like. And one of the 4 people I just mentioned will be there to provide answers to your questions as quickly as possible. In addition, this is going to be a recorded session. So if you want to refer back to any of the steps or processes that I've gone through, you will have this recording to refer back to at the end of our session today. Okay, so diving right into an overview of our platform, Benchling supports a wide range of R&D processes with applications that span your entire workflow, from design to experimentation to sample traceability and analysis. But each of these applications reside in a unified interface. So all of these applications can be accessed by scientists from a single point of entry in a common web browser, which is what makes Benchling so unique. In addition, Benchling's molecular biology suite is now the new standard for sequence design and visualization. So we offer a comprehensive suite of over 10 different molecular biology tools to handle all of your modern science needs, from CRISPR to digital experimentation analysis to large-scale batch analysis. But most importantly, Benchling's molecular biology suite is built with collaboration at its core. So you're able to share links, annotations, and feature libraries throughout your entire organization. Okay, so before diving into the platform, I am going to go over our projects and permission structure really quickly. You might be familiar with some of this information, but I'm going to describe how molecular biology kind of integrates with this. So as a reminder to everybody, projects are the highest level of organization within Benchling, and this is where permissions for your organization are set. Underneath projects, we have folders as well as sequences that can stand alone or be housed within folders itself. So your molecular biology sequences are going to live within projects. And because you may or may not have collaborators on these projects, you also allow collaboration on your molecular biology sequences. In addition, just to remind everybody, there are 4 basic permission settings. So the highest is the admin view, so you can view and create sequences in the project, as well as adjust the permissions of others. Next, we have the write permission, which allows you to view and create sequences, but you can't adjust the permissions of others. Third, we have our read permission settings, which allows you to view sequences. And lastly, we have our non-permission setting, which means that you cannot view or create DNA sequences within a project. So just to recap, not only does Benchling allow you to collaborate on your molecular biology, DNA, or amino acid sequences, but we also allow you to personalize how that collaboration happens by adjusting whatever permission settings your different collaborators have. Okay, so now we've gone over some of the introductory information, and so I'm now going to be going into our use case itself. So today I'm going to be modeling a sample use case in which I am a researcher that is part of an organization dedicated to breast cancer research, specifically mutations within BRCA. So similar to the previous boot camp, we will be conducting this training today from within the notebook, which is where the main application you are going to use to upload your experimental data and interface with our molecular biology suite. So as a researcher, part of this organization, the first thing I'm going to do is log into my Benchling tenant. So I'm going to go ahead and escape out of the presentation and then log onto my Benchling account. And just as a recap for everybody, all of the main applications of Benchling are housed on this left-hand side panel, and our workspace is on the right-hand side here. So the first thing I'm going to do is access a project that I might be assigned to work on. And so all of our projects are housed within this briefcase icon on the left-hand side right here. So I'm going to go ahead and click on that icon. And now I have a display of all the projects that I may or may not be a part of. And so I am going to navigate to the project put together for today. I'm going to go ahead and click on that project, and I see that a few files have already populated. And because I'm a researcher, part of an organization, they have put together a template or protocol that they want me to work through. So again, templates are a great way to standardize the way work is done through your organization. So I'm going to go ahead and open up that template now. So within this project panel view, I'm going to navigate to the top right-hand corner plus icon right here. And I'm going to go ahead and click on it and then scroll down to entry and then click on entry from template. And now I have the ability to select a template from this drop-down box right here. So I'm going to click on this drop-down and navigate to the correct template for me today, which is plasmid cloning of BRCA2 mutants. I can adjust the name if I'd like, make sure it's saving to the correct project. And now I'm going to go ahead and click on create. And from here, we can now see our template has opened up on the right-hand side within our workspace. So I'm going to go ahead and minimize this project view so we can maximize the template view. So I'm going to click on the briefcase icon again, and I see that my template populates through my entire screen. So today we chose a use case that should hopefully be very familiar to everybody on the call. So it's plasmid cloning of a gene of interest. And for me today, I'm going to be plasmid cloning a BRCA2 mutant. So just for some background information for everybody, BRCA1 and BRCA2 are tumor suppressor genes involved in DNA repair. So a mutation within BRCA2 usually results in a high likelihood of an individual developing breast cancer. So my study objective for today is going to be in vitro functional characterization of a newly identified BRCA2 mutant in DNA repair. So the experimental design within my lab might look something like this. So I have my region of interest within BRCA2. Today, we're going to be working with a mutation that is within exon 1 of BRCA2. And so the first thing I'm going to want to do is amplify that exon 1 region of interest with something like PCR. And then next, I'll want to perform a restriction enzyme digest of both that amplified region of interest as well as a plasmid backbone with whatever enzymes of interest I might have to make sure that they create correct sticky ends. And then once that digest has been performed, I'll want to ligate that region of interest within my plasmid backbone. And finally, because this is an unknown mutation, I'll want to compare that unknown region of exon 1 to either a wild type version of exon 1 as well as known mutations of interest and perform a DNA alignment. So I'm going to go ahead and exit out of this sample workflow now and scroll down through my template. And the first part of today's training is going to be dedicated to in silico visualization and design. So I'm going to be importing my BRCA2 gene of interest. Then I will be visualizing it with some of Benchling's molecular biology suite tools, and then finally translating that exon 1 region of interest to investigate further some of the biochemical properties that I might be interested in. So again, I'm going to exit out of this. And so our first step today is going to be importing the gene of interest. So I'm going to navigate back to that briefcase icon where my projects are held and make sure that I'm within the correct project. And I'm going to navigate to the top right-hand corner where the plus icon is, and then scroll down to DNA sequence and click on one of these options depending on how I want to import my DNA. So I'm just going to go ahead and click on new DNA sequence. And now I have the ability to import my DNA sequence depending on how me or my organization typically houses data. So because I have a file on my computer, I'm going to be using this Convert Files tab, and I'm going to be choosing a file from my desktop. So I'm going to click on Choose File, and then I'm going to choose this bracket2 file that I have downloaded from GenBank, and then scroll to the bottom right and click on Open. In addition, we do support files from FASTA, APE, Genius, SnapGene, Sequence Builder, etc. And when that upload has completed, you'll see this upload done green icon right here. But in addition, if you are using publicly available genomes on external databases, you can import them directly from here, such as NCBI BLAST. Next, you can import from directly specific chromosomal regions of organisms of interest. And lastly, you do have the ability to import raw sequences if you choose. But now that my sequence of interest has uploaded and the upload has been completed here, I'm going to click on Open Sequence. And now you can see that my sequence has directly opened up on the right-hand side of my workspace in a separate tab. So again, I'm going to minimize this project briefcase icon to maximize my workspace view. And Benchling immediately gives me the ability to visualize my sequence in a few different ways. So this linear map is the most consolidated kind of view of my sequence of interest. So if I want to see everything on one horizontal scale, I can view my linear map view. I can also choose to zoom in and out by toggling left and right through this minus and plus icon. Next, let's say I want a more granular view of my sequence. I can go ahead and click on my sequence map tab at the top left. And so here you can see the individual bases, exactly where annotations are adding enzymes and their specific cut sites here. Third, we have this metadata tab. And so this gives higher level information about the file within Benchling itself. So if I forgot who the authors were, I wanted to remember where this file was housed or when it was created. All of that higher level information about my sequence of interest is all within this metadata tab. And lastly, we have our description tab, which is where you and your organization can write down quick important information about your sequence of interest. So it's kind of like a notes or word attachment to your sequence. In addition, you'll notice that as I click between these different 4 tabs, they all populate as a whole screen view within my workspace. But let's say, for example, I want my sequence map on one side and my description tab map on the other side, and I want my workspace to be split. I do have the option to split and unsplit my workspace at the bottom right-hand corner of my Benchling screen. So here you'll see that we have this button called Split Workspace. And when I click on it, you can see that the different tab views populate side by side. And let's say I want my sequence map and my linear map both on the left-hand side. I can go ahead and drag my linear map tab to the left, and I can click between the two on either side to kind of adjust my workspace as needed. But I'm going to go ahead and unsplit my workspace now just by, again, toggling off that same split workspace button that I clicked on before. Right. So in addition, you do have the ability to customize what you are visualizing at any given point in time. So by clicking on this drop-down associated with the gear icon, I can choose to toggle on and off various features that Benchling supports, so annotations, primers, etc. Through here. And lastly, on the right-hand side, we have our molecular biology suite, which consists of 7 different tools that I will be going into more depth in as we go through this use case. But just to give a quick overview of it, at the top, we have our flag icon, which is our annotations tool. Next, we have the scissors icon, which is our digest tool. Third, we have this double-headed arrow, which is our primers tool. Fourth, we have this clock icon, which is our history tool. And I'm just going to give a little bit of information about that right now. So let's say I have accidentally deleted this one segment of interest, and I want to revert back to a previous version of my sequence. I can navigate to that clock icon, our history tool. And you can see that Benchling immediately auto saves all of the edits that happen to my sequence in the cloud without me having to perform extra steps. So this is extremely useful, especially if one, you accidentally make a mistake, or two, you just want audit logs of all of the edits that are made to a sequence of interest. So I can click on that previous version and click on reset to version. And an approval message will populate on the bottom left-hand side. In addition, you can edit the different version names. So if you're making very specific edits and you want to easily navigate between the different versions that Benchling has saved to the cloud, you can name them as such and easily again navigate between them. Third, you have the ability to clone versions to new DNA. So you can clone previous versions of your sequence within Benchling if you have edit forks that you want to edit. So all of these options are available through our history tool. Fifth, we have our alignments tool. So this is where you can compare your sequence of interest to other sequences in addition and see kind of how they vary by base. Sixth, we have our CRISPR tool, which allows you to analyze and create guide RNAs. And lastly, we have our information tool. And so this just gives you a snapshot into, again, that higher level information about your file of interest. So its name, where it's located, as well as the ability to download the file in whatever format you may choose, as well as archive it. So within the sequence itself, you also have the ability to manipulate the bases. So if I highlight a specific region and I wanted to change the bases or modify, edit, copy, I can right-click on a segment and choose between all of this different functionality that Benchling has to offer. And again, as we go through our use case, I'll go into this in more detail. But lastly, let's say, I have my mutation again, just as a reminder in exon 1, and because I'm going to be performing some experiments with that region, I want to know its length as well as, important experimental parameters. So if I select a region, which I can do by just clicking on that exon 1 annotation, or if I wanted, I could have clicked and highlighted that region. At the bottom left of my screen, some quick information populates. So it tells you what the start base is, what that end base is, which is base 192, how long that region is, as well as GC percentage and melting temperature. So again, very quick snapshot of experimental information I might need going forward. So now I'm going to navigate back to our template to recap some of the steps that I just took. So just to remind everybody, we first imported our file of interest from the computer and then explored different DNA import options. All of that was done through clicking on that plus icon, going to DNA sequence, and then importing that way, just as a reminder. Then we explore the different visualization features. So we reviewed the different tab views, which are up at the top left. We then toggle different features on and off with that gear icon. Third, we then explored the suite on the right-hand side. And lastly, reviewed those available editing options by selecting a region and right-clicking. So now we are going to begin exploring annotations. So let's say that, again, this file of interest is housed within a project that I have multiple collaborators on. And whenever those collaborators access this file, I want them to know that our region of interest is exon 1. So I want to create an annotation for exon 1 called region of interest. So the first thing I'm going to do is select or highlight exon 1. And I can do that very easily by selecting this already existing annotation. And I'm going to then right-click on that portion of the sequence and scroll down to create annotation. And now Benchling immediately takes me to this new annotation section of the annotations tool. So the first thing I'm going to do is give it a name. So I'm going to call it region of interest. The next thing I'm going to do is give it a type. This is a mutation in this case, so I'm going to designate mutation as the type. And by giving your annotation a type, you can eventually filter what types of annotations you want to view at any given time. And so once I create this, I'll show everybody how to do that. Then I'm going to confirm the position of this region of interest. So it starts at base 1 and ends at base 192. Lastly, I can change the color if I'd like, and then designate whether it's on the forward or the reverse strand or on neither. I'll keep it on the forward strand. And now I'll click on Save Annotation, and you'll see that region of interest has now populated right alongside exon 1. So our annotation has been created properly. And so I'm going to go ahead and click on that flag icon again, our annotations view. And so on the left-hand side, you'll see this option of filter visible by type. And again, I created region of interest with the type of mutation. And so if I go ahead and click on this, I'll see all of the different annotation types that already exist. So let's say I don't want my workspace to be very cluttered and I just want to visualize that mutation type. I can deselect these other types of exon and primer, and now we see that our sequence only populates with that region of interest as it's the only mutation type for right now. So again, this does give you the ability to visualize what annotation type you'd like to see at any given point in time. Okay. So creating mutations individually is very good for kind of one-off markers that you want to make. But let's say that for every BRCA2 sequence that I import, I have a large set of features that I want annotated onto it. And I don't want to manually have to annotate those features individually. Benchling allows you to create something called a BRCA2 feature library. So today I'm going to be adding a feature to a library that exists called BRCA2 features. And then I can auto annotate my entire sequence with items from that feature library to reduce a lot of the manual work that comes with individually annotating my sequence. So to access your feature libraries, the first thing you're going to want to do is navigate again back to that annotations tool and then scroll to the bottom right corner where it says edit feature libraries. And Benchling will take you to a different page. So if I didn't already have a feature library that's created, I can create this new library up here. But for today, we're going to be opening an existing feature library called BRCA2 features. And we're going to be adding a feature to it. So let's say, in addition to what I already have in this feature library, I also want exon 2 annotated. So I am going to give my feature a name. 2, designate it a type. It's going to be exon. Designate whether it's a nucleotide or protein because we're working with bases. It's going to be a nucleotide for today. And then I'm going to copy and paste the bases from my sequence here by selecting that exon 2 region and then on my keyboard hitting Command or Control C to copy it. And then pasting that region right here as the bases associated with exon 2. And now at the bottom right, I'm going to click on Add Feature. And we can see that now exon 2 has been added to a feature within this feature library here. And so now I've added all the features that I want. And the next thing I want to do is annotate my sequence with all of these features. So I'm going to exit out of this page. And on my annotations tool again, I am going to scroll to the bottom left corner of the pop-up and select Auto Annotate. And now Benchling gives me the ability to choose between one or more feature libraries. So I'm going to click on this box and then choose from this drop-down of feature libraries that exist. And so I'm going to click on the BRCA2 feature library. And Benchling is very smart. It gives you the different features within that library that can be annotated onto your sequence of interest. So here we see that all of these 4 features can be added if I'd like, but in this case, I already annotated my region of interest. So I want to deselect that feature from being annotated, but I want to keep exon 2, 3, and 4 as being annotated. So I keep those as selected here. And then I will go ahead and click on Add Annotation. And now we can see that those additional annotations have been added. So that exon 2 annotation and 3 annotation and exon 4 annotation. Okay. So the ability to share annotations through feature libraries saves a lot of time, and we would love to learn more about how you and your organization typically track annotations. So I'm going to be launching a poll question right now that should hopefully be visible for everybody on their screen. So the question is, how do you and your company currently annotate any sequences? 1, on an individual researcher basis. 2, shared annotations across teams and projects. 3, we don't annotate sequences at all. And 4th, we don't do any DNA or amino acid sequence work. Okay, so now everyone should be able to see the poll results. So it seems like the majority of everybody here currently annotates either A, on an individual researcher basis, or B, sharing annotations across teams and projects. So just to kind of go back into the Benchling platform. So not only does Benchling's feature library creation standardize, the terminology that you and your organization might be using, but in addition, our feature libraries do give you the ability to reduce the manual labor that goes in when individual researchers are perhaps going through and annotating manually every sequence individually. So feature libraries have a lot of value to them, and we hope that you and your organization get to use them to standardize your work, as well as, enhance efficiency when you annotate your sequences of interest. Great. So just to recap some of the work we just did with annotations. So we created a singular annotation in Benchling for exon 1 called region of interest. We then added a feature to the feature library, bracket 2 features, and then finally auto-annotated the sequence using one of our existing feature libraries. So the last thing we're going to do is explore protein translation within our sequence. So I'm going to navigate back to my sequence. And again, that mutation is within exon 1. And so the first thing I'll want to do is maybe overlay the translation that's associated with it to see what our amino acid sequence is. And next, I might also want to explore some of the biochemical properties that are associated with that eventual protein translated region. So if the first thing I want to do is simply overlay those translated amino acids, I'm going to create a translation within Benchling. So I'm going to select my region of interest again, and I'm going to go ahead and right click and scroll down to create translation, and I can choose the forward or the reverse strand. So we're going to keep forward, and now we see immediately that the overlying amino acid sequence is right alongside those 3-letter codons. And Benchling allows you to manipulate this at will. And so if I change those bases individually, so let's say I'm going to change this G to a T, and I click on enter, that associated translation is going to change alongside it. In addition, I do have the ability to change the amino acids directly. So if I right click on this amino acid and click on change amino acid, we see the different frequencies that are associated with a specific expression system. So this is something we call codon optimization. And so you can optimize or choose what amino acid you would like depending on the expression system that you want to fit. So you can choose between these different organisms and choose your associated codon depending on what you would like. But in addition to just overlaying this translation, I want to explore further the biochemical properties that might be associated with the entire translated protein. So I'm now going to select that region of interest again, and I'm going to right click and scroll down to create amino acid sequence. And I'm going to select the forward strand here. You can choose a new name, designating where it's saved, and then hit create. And now you see that Benchling has created an entire new entity that is associated with that region. So going over the top tabs on the left really quickly, first we have our sequence map. we can zoom in and out again. And so you can see those individual amino acid sequences. But more importantly, we have our biochemical properties tab. So again, I'm going to be performing some experiments with this region. And so it might be a gel or PCR, and I need a quick snapshot into that info. This biochemical properties tab gives me very quickly the molecular weight of that region, the isoelectric point, different extinction coefficients, and amino acid frequencies if I'd like. And we have that same description and metadata tab as before, but just wanted to highlight the difference between simply creating an overlaid translation within Benchling versus creating an entire new amino acid entity. So I'm going to scroll back to my template and recap some of the steps we just took. So we created that translated region of exon 1 by right-clicking and scrolling down to create translation. We then created a new amino acid sequence and explored some of the biochemical properties and went through how to manipulate those amino acid residues. Okay, so now that we have explored all of our visualization features, I want to start preparing my region of interest within the BRCA2 gene for PCR. So I'm going to go ahead and click on this workflow. And so for exon 1, we're going to be first attaching manual primers with the correct enzyme cut sites. So it's going to look something like this. We're then going to run a PCR with those primer cut sites added on to amplify our DNA. And finally, we're going to ligate our region of interest into this PUC19 plasmid backbone. So the first thing we're going to be doing is creating primers manually. So I'm going to navigate back to my BRCA2 sequence. And to create primers, I'm going to scroll to the right-hand side back to my molecular biology suite and click on this double-headed icon, which is our primers tool. So I'm going to click on my primers tool, make sure I'm on the primers tab, and then click on create primers. And I can choose between manual or wizard. Wizard is something we'll be going over in more advanced sessions. I'm going to click on manual here, and you'll see that this new design primers tab appears. So this is a place, again, where split workspace is going to be very helpful. So I'm going to go ahead and split my workspace, and I want my design primers tab on the right-hand side, and I want my sequence map on the left-hand side. So as I am designing primers on the right, I have the ability to visualize how that's checking out on my sequence map on the left-hand side. So because we are creating primers for a PCR experiment, I don't want to create a single primer, but a primer pair. So the first thing I'm going to do on my design primers tab up here is I'm going to select this single primers dropdown, and I'm going to select primer pair. And so now I have the ability to design both a forward primer as well as a reverse primer. And so I'm going to start by designing my forward primer. So I'm going to navigate to the left-hand side where my sequence map is, and I've decided that I want to create a forward primer that encompasses the first 20 bases of exon 1. So I'm going to scroll all the way to the left of my exon 1 region, and I'm going to click and, select all of these first 20 bases and make sure that the length is 20. And so again, that's where that bottom left length pop-up is very useful. So now I've confirmed that I've selected the first 20 bases here. And I'm going to go back to my right design primer tab. And there is a box under this forward strand that allows me to enter in the bases for my forward primer. So I'm going to go ahead and click on that box and then scroll to the top right where it says set from selection. And I'm going to choose to set from my forward selection. And so now we can see once I have made that selection, there's a shadow arrow that appears on the left-hand side of my exon 1, which gives me a preview as to where that primer would be held. And then the next thing I'm going to do is confirm that that 3' location is at base 20, which it should be at. And next, we're going to create the cut site attachment that we'll be adding to the 5' end of this primer. So navigating back to my template, it tells me what 2 enzyme cut sites I want to be using. So FSCI is going to be the 5' enzyme cut site for today. and PAC-I is going to be the 3' enzyme cut site for today. So we're going to start with FSCI. And so in this cut site box right here, I'm going to click on the drop down and type my enzyme name of interest. And so when I type up that enzyme name, Benchling immediately gives me the cut site that is associated with this region. So on my keyboard, I'm going to hit Command or Control C to copy the segment of 8 bases. And then I'm going to scroll back to the 5' end of my forward primer. And then I'm going to hit Command or Control V on my keyboard. And now I've added that cut site to the 5' end of my forward primer. The next thing Benchling wants me to do is specify what that overhang is going to be. So here it's 8 bases because that's how large our cut site was. And now our forward primer has been designed. So I'm going to do the same thing for the reverse primer now. So I'm going on my sequence map tab, going to scroll to the end of exon 1, our region of interest, and I'm going to select the last 20 bases. So again, I've selected that region of interest. I'm making sure that the length is 20 by checking that bottom left pop-up. And now I'm going to go ahead and click on the bases box under the reverse strand primer. And again, I'm going to scroll up to set from selection and then click on reverse in this case for the reverse primer. And I'll confirm that 3' location is supposed to be at base 173. And for our second cut site, it's going to be the enzyme PAC-I. So I'm going to type the enzyme to get its associated cut site. And from here, I'm going to Command or Control C the set of bases, and again, add it to the 5' end of my reverse primer. So here now I'll designate the overhang as 8 bases again. Benchling has highlighted the region that's going to be attached to exon 1. And the next thing I'm going to do is now check the secondary structure of both of these primers. So once I click on that button, I can make sure that all of the parameters align to whatever me or my organization want it to. And finally, give names to each of these primers. So I'll call the forward primer, primer 1, the reverse primer, primer 2, and make sure that they're saving to the correct location, and then click Save Primer Pair. And now I'm going to unsplit my workspace so everyone can see this further. And you'll see that I have created primer 1 that has attached to the first 20 bases of exon 1, my region of interest, and we have primer 2 that's added to the last 20 bases of my region of interest. So we've now manually created the primers, and the next thing we're going to want to do is run a PCR. So you can again navigate to the right-hand side to that double-headed arrow primers tool. And now on the pairs tab, you can scroll to the bottom left-hand corner where it says create PCR product. Go ahead and click on that option. I'm going to keep all of the default boxes that Benchling has checked and click copy. Make sure it's saving to the correct project. And now I have created my PCR product. So just to zoom in a bit so everyone can see, we again have that forward primer that has the first 20 bases that's annealed to exon 1 and this FSCI cut site of interest that's been added. And now we also have primer 2 that's saved to the end region of exon 1 with our PAC-I enzyme region of interest. So this PCR ran as we'd like, and we now have the appropriate 2 enzymes as the sticky ends for our region, preparing it to be ligated within our plasmid backbone. So now I'm going to navigate back to my template. And so we have created the primer manually with our upstream cut site at FSCI, our downstream cut site at PAC-I, and we've now run a PCR reaction. So for right now, I've had this PCR resulting sequence, and I want it to be easily mentioned or accessed by individuals within my organization. So this is going to open up our next polling question, which I'm going to launch right now. So our question for everybody is, how do you currently reference a DNA or amino acid sequence within your notes? So the first option is copy and paste the sequence as text. The second option is copy and paste the URL link to the sequence. The third option is download the sequence file and attach that to your notes. Fourth is @Benchling a Benchling sequence, and fifth is other. Okay, I'm going to go ahead and end the poll now and share the results with everybody. So actually it looks great. Seems like the majority of everybody that voted actually mentions a Benchling sequence. But for individuals that currently download a sequence file and attach it to your notes, or copy and paste the URL or entire sequence as a text, you probably know that that can really clutter up your workspace, as well as take up extra data and storage on your laptop or whatever file you're using. So I'm now going to demonstrate mentioning within your Benchling notebook. So within Benchling, for those of you who don't already know how to do this, you have the ability to directly link your sequence or file of interest by simply typing in the @ symbol, as well as the name of whatever file you're interested in linking. So for today, we're going to be linking our PCR product of interest. So the name of that file is BRCA2 1 through 192. And so now I've created this two-way link between my notebook entry and my PCR product. So if I go ahead and click on that link, it immediately allows me to navigate to that PCR product. So again, very helpful. One, if I want to save storage and not clutter up my workspace, but more importantly, if I have a lot of data and I want to easily be able to navigate through all of it, @Mentioning is an incredibly helpful tool for me and my organization to be using. All right, so the next thing that we're going to do is, now that we have our PCR product of interest with the correct sticky ends, we want to ligate that into our plasmid backbone. So I am going to be opening up a PUC19 plasmid that has our restriction enzyme cut sites of interest by going back to my project icon and then scrolling down to my PUC19 sequence and opening it. And so just as a point of reference for everybody, when you do have circular or plasmid DNA within Benchling, you do get this additional plasmid tab. So you can view it with this topology. And just to make sure, we have that FSCI cut site right here, as well as that PACI cut site down here. So within this region right here is where I'm going to want to add my exon 1 region of interest from my PCR product. So today I'm going to be going through a copy and paste vector assembly, which is very useful if you already know what your two enzyme cut site ends are and want to copy and paste that region in directly. So I'm going to navigate back to my PCR product, and I'm going to zoom out just a bit. And so I'm going to first select that FSCI cut site on the left-hand side, and then I'm going to hold down Shift on my keyboard. And then I'm going to click on PACI, which should be the right flanking end of my region of interest. And then on my keyboard, I'm going to hit Command or Control C to copy this region of interest. And now I'm going to go back to my plasmid backbone tab, and I'm going to click on that FSCI left cut site. I'm going to hold down Shift on my keyboard again, and then click on the PACI cut site on the right-hand side. And so I've now selected the region where I want to insert that region in, and I'm going to hit Command or Control V on my keyboard to paste that region in. And now we see that it has assembled correctly. We can double check that because we have those two annotations that we had created for exon 1 region of interest. And so this is the easiest way to kind of create a recombinant vector, especially if you know what parts should be fitting together. But we know that this is not the only way in which you and your organization might be creating assemblies. And so we have our third and last poll question for everybody, which I'm going to launch right now. And our last question is, what is your typical construct design approach? So first, restriction-based cloning; second, Gibson assembly; third, Golden Gate; fourth, simple copy and paste; and 5, other. Okay, so it seems like the majority of everybody uses either restriction-based cloning as well as Gibson assembly, and some use Golden Gate and simple copy and paste. And so while today I only went through this copy and paste method, just for everybody's reference, we do have more advanced levels or types of assemblies that are available for you and your organization to use. So at the bottom right-hand corner, next to that split workspace icon, we have our assembly wizard. And if I click on create new assembly, I have the ability to create a digest and ligate assembly, Gibson assembly, or golden gate assembly if I choose. Okay, so just to confirm everything we did, we viewed the plasmid and the associated cut sites. We then copy and pasted our PCR product within those cut sites and have now explored the assembly wizard. So the last thing we're going to do now is perform a DNA alignment. So again, we have our unknown mutation of interest and we want to compare it to a wild type and maybe 2 other known mutations of interest. So I'm going to navigate back to my PCR product of interest that has that unknown mutation. And so I'm going to scroll to the right-hand side, our molecular biology suite, and click on the alignments tool now. And so from here, I can click create new alignment, and I can either choose files from my computer or choose sequences that are already uploaded into Benchling. So I already have a few sequences I'm going to choose that are within Benchling. So first is our wild type. I also have a mouse mutant and a human mutant. And now we see all of the sequences that we want to align or compare to each other. And right now, our PCR product is the template strand, but in this case, I want exon 1, the wild type, to be my template strand. So if I simply click on that sequence, it immediately changes that to be the template strand for today. And I'll keep this MAFFT algorithm, and then I'll click on create alignment. And now Benchling has aligned the 3 secondary sequences to my primary wild type sequence. So at the bottom, we have this mini map view of our entire alignment. And so this is where you can adjust in how much depth you are viewing your alignment in, as well as where in your alignment you are. So if I wanted to view this in more depth, I can minimize this viewing window. And we see that it's much more zoomed in. And if I drag and hold that viewing screen and kind of scrub through the entire alignment, I can move back and forth to whatever region of interest I'm interested in viewing. In addition, at the top, we have this ability to find mismatches directly. So I can click on the left or the right arrows, and Benchling will immediately take me to the next mismatch. But finally, we have our voting system at the top. And so, for example, if I hover over this column, it tells me that there are 3 votes. And so here I can see all 3 of my secondary sequences are aligning to my wild type sequence. But for example, if we scroll to this region right here, we see that only 2 of our secondary sequences are aligning to our wild type sequence because this variation or sequence doesn't. So alignments tool is very helpful for quickly referring to regions that might have more or less annotations, as well as individually going to each differing region between different bases. So again, very useful for cross-comparing sequences to each other. Right, so that concludes the in-application portion of our demo. So I'm just going to go back to our slides and give a few product updates. In addition to next steps that everyone can take. So sorry, give me one sec. Okay. So some of our new molecular biology functionality. So we now have introduced bulk codon optimization. So again, if you want to optimize the amino acids you're using to match the frequencies of a certain expression system, we now have the ability to do that through bulk codon optimization and adjusting those parameters in the description tab. In addition, we can now copy to for amino acid sequences, export annotations with multi-part GenBank files, bulk back translation updates, but for a full list of all of our product release and new features, you can go to help.benchling.com and get a much more comprehensive list. In addition, we would love to get your feedback. So if you could take the next minute or so to complete the survey found in the chat, that would be great. Okay, assuming that everyone has generally finished that up, just some resources for everyone to access. So we have a very large repository of help articles on help.benchling.com, which can answer some of your frequently asked questions, as well as go through some major processes and steps that other organizations have needed help with in the past. But if you don't find your answer within one of our help articles on help.benchling.com, we also do have friendly in-app chat. So you can be connected directly to somebody on the customer experience team, and they can help you troubleshoot issues, as well as answer any questions you might have. But in addition, if you do have additional training needs, please feel free to reach out to the training team at training@benchling.com, and we will get back to you. But thank you so much, everybody, for attending our bootcamp today, and hope to see everybody at some of our more advanced sessions in the future. Thank you.